All right. Can you guys hear me? It's gonna work. Admit. All right, can anybody see me or hear me? So, um, Um, record button. Can you guys hear me at all?
Yes.
Oh, thank you. Oh, it's working. Okay.
Yep, can see your screen and we can hear you. Well, I can.
Okay, I just need to find the record button, which was here earlier.
It says it's recording.
Oh, does it? Okay. All right, cool. All
At
right.
least I think so. I see a recording going on.
Okay. So I thought it was recording the minute I opened it. All right. So I guess I'll give everyone a few more minutes. I've got like 7 of you on. So I just want to try this out because it seems to be easier on my end than WebEx. So you guys can see me okay?
Yep,
Okay. All right. Who's talking?
Catherine.
Okay. Thank you, Catherine.
You're welcome.
I was trying to figure out how to unmute myself on this. I've never used it before.
Yeah, I guess from here I can mute you guys or ask you to unmute, or I can turn your video cameras on or off. See, Robert, you're on, but I can't see you. Like it literally says your video camera's on, so it's something I gotta just play with. But this will work. All right, so if everyone's okay, I want to just go over. I think this is going to be chapter 14 tonight. Hopefully we can end around 6 or 6:30. This shouldn't take that long. And then we'll get into 15 and 16, which are a little bit more difficult. Okay, everyone's okay with that?
Yep, sounds good.
All right, I also uploaded a practice little test in Lab. If any of you have Lab with me, I uploaded some videos that I did in March, right after COVID hit. So they're there also. And then what I'll do is I guess I'm stuck with giving everyone like a midterm and a final, and that's going to have to be your lab grade. So I'll be asking you guys some theory. I'm going to try to go on Thursday and do a street plate demonstration and see what I can put together and videotape on my own with my iPad. So I'll try to throw it together. Otherwise, I have to put up somebody else's YouTube or Khan Academy videos, which I'll do. I just, like I said, I feel really bad that you guys aren't in lab. You guys probably aren't upset about it, but the lab part is the super fun part about micro, actually growing it and seeing it and inoculating it. So I apologize that you guys don't have that opportunity. But I'll teach you what I can, all right? And we'll get you through your board exams or NCLEX or whatever you need to get through, all right? So let me make sure I can get this working. Okay. All right, so I want to go over the immune system, and I'm going to spend a little time on it, probably more than most any other instructor you have. But I've talked to Dr. Lucido and a lot of my friends and MPs and nurses, and a lot of my students have sent me back. My NTRIP students and some of my ECC students have told me that once they got into nursing, that it was extremely helpful, and the immune system and pathophysiology were all reviewed for them. So I'm going to stress what I think is important. If you guys have any questions, please ask. It's really hard for me to not ask questions. I feel like I'm in a straitjacket. So I will ask you questions. And please realize if I'm asking you questions, you're probably going to see some version of that question on an exam. All right. I also was up last night. I had to redo the exam 3 times because every time I went to do it, it would go away. So if someone could just try it, or at least make sure it's there, you don't have to hit the OK button and let me know if there's a problem. I think it's going to be due Thursday. If anyone has a problem, let me know. I can certainly change. It's not a big deal. All right. So I want to talk about the defense mechanisms of the host. So, we're making sure that we talked last week or this week about our skin and integumentary system, tears, mucous membranes, things like that were boundaries. All right. So we're also going to be talking about once something breaches those boundaries, just because it breaches those boundaries doesn't mean you're going to have an infection. And you could have things growing or multiplying, and until they start causing a problem or causing inflammation or disease state, you're not going to be affected by that, right? Well, the difference between innate natural defenses, these are present at birth, provide non-specific resistance to infection. So these are going to be your macrophages and your monocytes and pretty much your neutrophils. They're going to attack anything that seems foreign. And then we'll start really talking about the adaptive immune system, which is much more specific, has to be acquired. And by acquired, it means you have to be exposed to an antigen, all right? And then we'll be talking about B cells who wait their whole life to be exposed to a specific antigen so they can express their ability to make a specific antibody. And we'll talk about our T cells. The T cells literally have to be introduced to the antigen by a helper T cell and B cells so they know exactly what to target. We'll talk about that in chapter 16. All right. So your first line of defense is innate. It's non-specific. All right. So these, like I said, are your physical barriers, skin, tears, coughing, sneezing, chemical barriers, the acidity on your stomach, the acidity on your skin, low pH of your stomach, lysomes. If anybody remembers what those are literally in your tears. All right, in case I forgot to mention, I'm pretty sure I did, lysomes are in your tears. They literally go through and they work like penicillin. They go in and break all those building blocks that hold that peptidoglycan layer together and gram-positive bacteria. Your digestive enzymes, either in your stomach or the ones that are released from your pancreas into the duodenum. And we have some genetic barriers. Some people are, more genetically prone to a disease or not prone to it. It really depends on your resistance to genetic makeup. So it depends on how your genes or epigenetics are shuffled. When we start talking about B cells and T cells, we're going to find out that your body literally takes a bunch of your genetics and they run it like like a lottery, like a quick pick, and they literally make tens of thousands of different combinations of B cells, and they're just waiting their whole life to be exposed to one specific antigen they've never been exposed to before. So your body is so incredible, it can make all these B cells that just are waiting to come across something that triggers their ability to make an antibody to it. And once they do that, they make antibodies and they make memory cells. So in case we're exposed to it again, we already have the recipe and we can make those antibodies immediately. So your first exposure with an antigen, it can literally take your body 5 to 10 days to make antibodies to have a big enough titer in your blood supply to start affecting it. The cool thing is if you're exposed again, all right, it takes 24 to 48 hours to bring that titer right back up above where it was before, and now you're able to attack that pathogen or specific disease much quicker. And the other cool thing is those antibodies stick around for a lot longer. They're making sure that there's no leftover antigens anywhere.
Hey, Dr. Phil.
Yes.
Kathleen and Anthony said they're both trying to get in and they're getting that message that says they're waiting and that you let them in soon.
I'm sorry. I said admit everybody. I am so sorry. Thank you.
Oh, you're welcome. They just asked me to
admit all. No problem at all. Thank you so much. This is all new to me. Looks like Lindsay wants to get in. Look at how popular we are. They all want to get in. All right. Okay. So it looks like Isaac's in. It's Robert. Kathleen is joining. Thank you, whoever just helped me out here.
You're welcome.
More. I'll figure this out. Uh-oh. Okay, so second line of defense are your phagocytosis, inflammation, fever, and interferon. All right, now, second line of defense is innate. All right, so that means that you don't have to think about it. It's already there. You're born with it. So phagocytosis. So a lot of your macrophages, if you're not familiar with that, your macrophages come from monocytes. Monocytes leave your bloodstream. They get into your interstitial fluid. Those become macrophages. Macro meaning big, phage is to eat. So these things, well, they're huge eaters, and they literally eat. They'll engorge themselves until they literally blow up, right? Inflammation, we'll talk about that a little bit. Your body has the inflammatory process to flush out things, and we want to be able to bring more nutrients if there's damage. We'll talk about that. Fever, I mentioned earlier, your body's going to have fever. It's going to raise your temperature, right? And people may think that that's annoying, but remember, we talked about those bacteria, and they only have a certain level of temperature they can live at. So if we raise that up above what they're comfortable with, they're not going to replicate very quickly, right? And the other really cool thing about fever is it increases your temperature, it increases your metabolism, and it puts your immune system on high alert, revs it up quite a bit. And interferon, I don't know if anybody remembers from yesterday, we talked about interferon, all the cool things it did. And once again, especially if we have a viral infected cell, this will generally go and tell that cell to create or perform apoptosis. It's telling it, please kill yourself because we don't want you to be a replication factory. Right? And the third, which is the coolest and probably the most difficult, is your acquired immune system. And the big word here is very specific. All right, so B cells are only going to make antibodies to certain antigens, right? And T cells, if they're functioning properly, unless we're talking about a type 4 hyperreaction, they will only target cells that have a very specific antigen, which they are told about. The B cell and the T cell have to agree. They have to sign a contract. They have to handshake on it, literally. And then when that T cell comes across any cell that has an antigen, it goes in, it literally stabs a hole in it and sort of pumps a whole bunch of digestive enzymes, kind of throws up inside of the whatever it is and kills it. It's kind of awesome. All right. If anyone else tries to get in, just let me know and I will let them in. All right, so to protect the body against pathogens, the immune system relies on a multi-level network of physical barriers, immunologically active cells, and a variety of chemicals. And we'll learn about a couple of these chemicals, which are relatively important. So once again, your first line of defense is any barrier that blocks invasion at portal of entrance. So these are non-specific. And like I said, these are your keratinized protein of your skin. Mucous membranes would have immunoglobulin A in them. And I mentioned yesterday about right now, I'm a little concerned about I don't want to use the word overuse of hand sanitizers, but people are using it like crazy. So I'm just a little bit concerned about them breaking down the integrity of the skin, and with all the alcohol in it, unless it has glycerin in it, we're going to start drying it out, and we can provide little cracks and crevices for bacteria to start multiplying. And if that breaches into your basement membrane or your bloodstream, hopefully it won't be a big issue, but it can potentially cause a problem. All right, second line of defense, we said were protective cells and fluids, inflammation, and phagocytosis. And third line of defense is very specific, and it's acquired. All right, it's a little bit on the adaptive side. Acquired with exposure to foreign substances, produces protective antibodies, and creates memory cells. It's very specific. All right, so please remember, this is really the basis of any kind of vaccination. So with vaccination, I have my own thoughts on that, which I will keep to myself, all right? But with vaccination of any type, we're literally taking parts of a capsule of a bacteria or the antigenic part or antigen from a specific organism, and we're purposely introducing it into the body so that your dendritic or your antigen presenting cells will take those antibodies to your lymph nodes, And I asked you guys yesterday, why would your lymph nodes swell? We talked about how if they're swelling, you have a antigen antibody reaction going on in there between B cells and possibly T cells. So if those B cells come across an antigen that we purposely put in the body, your dendritic cells or antigen presenting cells, which are around your periphery of your body, they'll literally grab that, digest it, process it, they package it nice and neat, and they literally carry it to your lymph nodes, right? And they present it to your B cells, hoping that your B cells will recognize that. And then your B cells get super excited because they've waited their whole life for that one antigen that they can make antibodies to. And then they make antibodies, and it says right here, they create memory cells. So we make memory cells, we make more B cells, make daughter memory cells. And then whenever we expose that antigen again, these memory cells already have the recipe in their card catalog. They pull it up and they just start pumping out antibodies. And if the video plays in Blackboard, these B cells literally, once they're activated, can pump out like 10,000 antibodies per second into your bloodstream. It's absolutely incredible. All right, so here's just some physical barriers. I'll be asking you guys a question similar to this on the exam. You might get that in your question block or whatever. So once again, on your exam, I'm going to take like 20 to 40 questions, do a block, and then I'll just say pick 8 from that, 8 from this chapter, 8 from that chapter, and give you a whole varied chunk of them, right? So your skin and mucous membranes are the respiratory, urogenital, eyes, and digestive tract. And remember, once again, please try to keep in mind anything in your body that has a mucous membrane. You can argue all you want. It literally is the outside of your body. So it's an outside surface. Anything that wants to get in or digested or into your bloodstream or literally lungs, urogenital tract, it has to get through this mucous membrane. And once again, there's IgA in there. So a lot of things can't get by it. So the IgA will actually bind to it, make it so big that your second line of defense, your phagocytes or monocytes, or sometimes even your neutrophils will say that is big. It doesn't belong. There's something going on. You've attached a big IgA molecule to it. So it'll go in and just engulf it and try to kill it, right? The outermost layer of your skin is composed of epithelial cells that are compacted, they're cemented together, and impregnated with keratin. So please remember, when you're looking at someone, their skin, their hair, that's all dead. Keratinized protein pillows, remember, from your basement membrane, as things go up, they become less and less alive. And we kind of want the skin on the outside of your body to be dead, right? We don't want it to be alive because we're braiding it off all the time, creating dust and whatever it happens to be. So we want that to be dead. And we kind of also don't want it to be a really good food source for some kind of fungus or parasite. Right. Few pathogens can penetrate it if it's intact. It's when we have cuts or lacerations or the hand sanitizer, they can get into that, so that's not great. We have the flushing effect of sweat glands, and please realize your sweat glands are releasing some toxins or kind of flushing out the body. They might be releasing some acidity, some salt, things that would make most bacteria wouldn't be the most advantageous place for them to live. Right? Linking and tear production are releasing those lysomes, which are going to break apart all of the peptidoglycan layers of your bacteria. And then our stomach acid, which kills a lot of bacteria when you're swallowing your own saliva. It's full of bacteria. Right? Or if you eat something that might be semi-spoiled or something might be replicating in it, we want that stomach acid to kill it. The problem we're going to come across is when we start engulfing cysts or larvae or things from other protozoan or other things that we're swallowing those. The stomach acid actually goes in cysts, cysts, so it breaks open that cyst. And now we've kind of cracked the egg open on whatever that organism might be. But we'll talk about that in a few weeks.
Can you hear me?
Yeah. Who's that? Robert?
Yeah, I finally got it working. It was on my end.
Oh, okay. All right, cool. I can't see anybody, but I'll figure that out, I guess.
I asked a question. Let me pull it back up. How does the immunity break down after you receive a vaccine? Like, say, if you got it 20 years ago, how does that, and you do a titer now?
Yeah, so you're going to do a titer, and your memory cells are, theoretically, they're supposed to be good forever. But, yeah, after 10 or 15 years, they may not be as effective, or your titer might be, you'll still have the antibodies to it, but your memory cells are probably depleted, so they wouldn't be able to produce a full-on war for that. So like I said, last week, I got that pertussis, diphtheria, tetanus shot. So, that's something you want to do every 10 years. It'll be interesting once we get these, these COVID vaccines, but the problem is people that are being tested now, as far as I understand, if you've been exposed to any coronavirus at all since SARS, which was actually more pathogenic than what we're dealing with right now, they're saying that you could test positive, and not falsely positive for that. But yeah, we might say in 5 years, you no longer have antibodies to COVID or whatever. So hopefully the governmental agenda isn't to force vaccines on people. We should be able to have a choice, but we'll see. But yeah, usually the memory cells don't last forever. Good question. Did that answer it, Robert?
Yeah, thank you.
All right, good. All right, mucus coats. Mucus coat impedes attachment and entry of bacteria. Nasal hairs trap large particles. And I'm sure you've been told that they do the ciliary respiratory escalator where they move things up your throat. We'll learn about a couple things in the end, like whooping cough and diphtheria. They can go in and they can disrupt the cilia, the motion of it, so the cilia gets very confused, and your goblet cells just start releasing all this mucin. All right, and when that comes in contact with the fluid of your throat, it turns into mucus. All right, and then we have these exploding goblet cells of mucin turning into mucus all over, and there's no way to bring it up towards your throat so you can spit it out. All right, so if anyone has ever heard diphtheria, all right, their cough, the children, it affects the children, they're coughing and hacking because they can't get the mucus out of their throat, and what they do is they cough, and if you don't know the cough mechanism, your diaphragm contracts really quick, and it releases like an arrow out of a bow and arrow. It explodes real quick, and it takes all that air and shoots it out of your lungs, hopefully that you can propel whatever's in your throat, whether it's an irritant or mucus, out, so you can spit it out or swallow it or whatever you do with that, whatever your projectile or whatever you're releasing from your respiratory system. Okay. Now here's just some non-specific chemical defenses. Sebaceous secretion. So if you're not sure what that is, that means sweat. Your sebaceous glands, your sweat glands, all right? And believe it or not, there are people who don't sweat. There's a certain percent of the population that does not sweat. All right, you might think that's cool, but they're gonna overheat, and they can't release as many toxins, and they generally will have more things growing on their skin. Right? Lysomes is an enzyme that hydrolyzes the cell wall of bacteria and tears. All right, same thing as we said before, the peptidoglycan. It kind of chiseled out that cement that holds those cinder block type structures together, those amino acids, the nano-nag units. Right. High lactic acid and electrolyte concentration in sweat, skin's acidic pH, hydrochloric acid in the stomach, digestive juices and bile in the intestines. Semen contains antimicrobial chemical. The vagina has an acidic pH, except during certain hormonal conditions, it may change the pH, and then we have issues with opportunistic infections and glycogen production, and sometimes you can have some kind of albicans, Candidus albicans growing in there. All right, so genetic defenses. Some hosts are genetically immune to diseases of other hosts, right? So I mentioned before that my family really gets sick. I've never literally ever had the flu, and I cannot remember a day when I was actually sick in bed. All right, so my brothers and I and my mother never get sick. So genetically, I guess we won the lottery, or we take good care of ourselves. I don't know. But I also know people that are constantly sick from the smallest things. So either their immune system is being compromised, or it's doing a whole bunch of it's after a whole bunch of other things, and it can't be as effective as others, right? Some pathogens have great specificity, and this isn't really pathophysiology, but there are some diseases or some pathologies that are more prevalent in different nationalities or races. So genetically, their epigenetics or their immune system, they may have a greater propensity for that. And remember, some of these organisms really are very specific to certain cells or certain organs in certain bodies, and they tend to, there might be a, even if it's a 10 or 12% increased rate due to certain races or genetics, that's possible. We also know that people with O-type blood, they have more incidence of H. pylori, and they theoretically get stung by more insects than people who don't. But, they're saying, well, insects are attracted to the carbon dioxide you're exhaling, but they're saying people with O blood types statistically get stung a lot more. So I don't know what that all means. All right, some genetic differences exist in susceptibility. So here's some structures, functions, and organs of the immune system. Right. So the study of the body's second and third line defense is called immunology. And like I said, a lot of instructors tend to kind of breeze over this or don't spend a lot of time on it. Unfortunately, you guys have me, and I think it's very important, so we're going to spend a little time on that. Functions of the healthy immune system. All right, so you see this thing here, it's called PAMPs. These are pathogen-associated molecular patterns. We'll see that in a minute. So your white blood cells or some of your macrophages, monocytes, they will go on to look for pathogen-associated molecular patterns. So they may not know that what specific bacteria it is or what specific organism it is, but certain organisms, their genus or species are close enough that their antigens all have a similar pattern to it. So I could say an example, you could be walking down the street, all right, and you may not know jack about dog breeds, but you would at least know it's a dog, or you wouldn't know anything about cat breeds or horse breeds, but you are able to notice it. So these white blood cells, they kind of notice something that isn't quite right. They don't know exactly what it is, and they'll just go in, detection and recognition of foreign cell or virus. They go and kind of attack it. They try to destroy it, and they'll try to present it if they can to the immune system. So these are surveying the body, recognition of foreign material, destructions of entities deemed to be foreign. Right? So like I said, it could be bacteria, it could be protozoa, could be helminthes, a worm, even a tick or, a tick. They'll notice that in the skin, and they'll know it's something's wrong, and they'll try to at least attack it. Or with chemotaxins, they'll send out chemicals that are telling the immune system, listen, I got something in quadrant A over here, I need you to send somebody else. It's out of my scope of practice, they'll send somebody else. Right. And these white blood cells, they realize that these pathogen-associated molecular patterns are their own, so there's no reaction. But the minute they come in contact with something that's foreign, all right, they're going to go in and attack it and engulf it. So the immune system, large, complex, and diffuse network of cells and fluids that penetrate in every organ and tissue. So we're going to see a picture up here of your capillary beds, and then outside your capillary beds, you have interstitial fluid, and your cells are all floating around this interstitial fluid, and that's how we feed our cells. Things diffuse out of the capillary beds into the interstitial fluid with, our glucose, amino acids, blood cells. And then as the pressure builds, the fluid goes back into the immune system by oncotic pressure. And I mentioned the other day, when that fluid gets really, really elevated, those lymph levels will open up. The fluid will go into your lymph nodes. They'll close off, and then once again, these are going to your lymph nodes. On the way to your thoracic duct on the left, or your right subclavian, or yeah, thoracic duct on your left, right subclavian on the right, and we want to bring those fluids back to the right atrium. We want to recirculate those. All right, so we're going to see a picture of the reticular endothelial system. All right, whenever you see endothelium, you should be thinking about the inside of a blood vessel. It's always endo is always inside. So reticular endothelial system. So we're going to see these molecular organisms, white blood cells, antigen-presenting cells, are all floating around. They're just waiting to see if there's anything foreign. And I said these are in the basement, by the basement membrane or by the capillary bed. So let's say you got caught, stung, or whatever. Something was able to breach your skin or get around your organs. They're going to be in that interstitial fluid. We want these things floating around. They're like little sentinels figuring out if there's anything foreign. They want to alert the immune system to it, or bring them to lymph node or something so they can get some recognition. We have extracellular fluid, interstitial or extracellular fluid. Any fluid, I'm not in a cell, is extracellular. Interstitial fluid is a fluid that bathes all your cells. All right, we also have stuff in the bloodstream. You want all these things circulating in the bloodstream, right? And we'll, later on, we'll go through a CBC differential. I'll list all the cells, and we'll know what they're, if they're elevated, what they mean, right? And then we have our lymphatic system. Right? So a lot of people seem to skip over the lymphatic system. They don't think it's important. So please realize a lot of your immune system is in the lymphatic system. And I want you guys want you to realize too, no matter what you were told before, 60 to 70% of your immune system up here literally lives in your GI tract, all right? If you remember Peyer's patches or any of those molecular associated lymphatic system, right? They live in places other than your lymph nodes, and they live in your GI tract. All right, so here we are with the immune system definition. So our white blood cells, leukocytes, and we saw this yesterday. I said site was a grown cell, leuko means white. And remember, leukocytin, Staph aureus can produce it. So it'll just kill these things. It comes across it. The innate capacity to recognize and differentiate any foreign material. So your white blood cells, if they're working well, will recognize anything that's foreign. So if you have a CBC with differential and your neutrophils are elevated, right? Neutrophils, usually that indicates a bacterial infection. So neutrophils they're always the first cell on site. They can attack things, and they actually can become antigen-presenting cells if they need to. It's not their primary job, but they can. They're pretty multifunctional. All right. So non-self is any foreign material that's non-self. That should trigger your white blood cells or immune system. So it can be, it can be bacteria, it can be virus, it can be protozoan. It could be a splinter. It could be dirt. It could be a piece of glass. It can be anything in your body. It's foreign. Your cells should recognize it. That doesn't look quite right. All right, self is anything, normal cells in the body. And then I mentioned earlier, pathogen-associated patterns. Pathogen-associated molecular patterns. Molecules shared by microorganisms. So they're going to have some general patterns that your white blood cells are going to recognize innately. They don't even have to be told. Okay. Pathogen recognition receptors are receptors on white blood cells for pathogen-associated molecular patterns. So these are patterns on microbes, and then your white blood cells that have these pathogen recognition receptors. So they are just saying, well, we have receptors on our white blood cells that recognize patterns or pathogens, and they're actually recognizing these pathogen-associated molecular patterns. All right, so here's a picture of your reticular endothelial system. All right, so here's your bloodstream. Remember, everything, every cell in your body has to get nutrition. Even your adipose tissue, if they don't get nutrition or oxygen, to become hypoxic. We can have ischemia. things won't be able to replicate. They won't be able to function. Metabolism slow down. It's not a good thing. So these are floating around outside your capillary bed. This is all extracellular fluid. And this right here is literally going into the capillaries. I said when this pressure gets big, these valves, they kind of open up. When this pressure equalizes, the valves shut. Now, when you move, I should have told you in A&P or biology or whatever, these are going to go towards your lymph nodes. And remember, their final destination is your left subclavian or your left thoracic duct or your right subclavian. And these are always moving towards your heart. Their goal is to get to the right atrium. All right, these are reticular fibers. They kind of hold things together. And if you saw these in A&P with the cats, these would looked like very thin white tissue paper, membranous things that you saw when you pulled it apart. They're literally there. They're kind of like a gauze. They're kind of holding the material together. Very, very pliable, but they're really holding things together. Right? And then here are your white blood cells. They're just, you know, floating around out here. In case anything breaches this, they're going to go in and grab it. Right? So if any of you have ever had a massage done, and a professional massage, remember they're always, when they're massaging, they're always going from distal to proximal. They're always going from far away towards the heart or the center. So they're literally, when you have a massage, they're massaging all your tissues, but they're really flushing out your lymph nodes, getting rid of all that tissue, right? Trying to get all this fluid in your lymph nodes. And they should tell you at the end, you know, rest, drink lots of fluid, because we just flushed out your lymphatic system. It's going to your right atrium. We're putting that in your bloodstream. So now your kidneys have to really flush all that out. It's a good thing, right? But your kidneys might have to do a little extra work for the next 24 hours. Right? So here's that system again. It's a network of connective tissue fibers that interconnects other cells and meshes with the connective tissue network surrounding organs. This really, like I said, I want you to think of it as gauze or something that's tissue paper that's really holding things together, right? And in here we see our macrophage, right? Those are big eaters, right? Neutrophils are going to recognize any kind of foreign material, right? And there's our dendritic cells, all right? So we're going to learn later that your dendritic cells are literally antigen-presenting cells. So anything that these pick up that are antigenic, they're going to present it to the dendritic cell. It's going to recognize it as foreign. It's going to process it. And this guy is going to literally go to your lymph nodes and carry it to the lymph nodes. When you go to your lymphatic system, open one of those valves open, go in. It's going to crawl its way to your lymph node, and it's going to present it to your B cells. All right, inhabited by phagocytic cells, mononuclear phagocytic system right here. Macrophages are ready to attack and ingest microbes that pass the first line of defense. So they're just sitting here waiting for anything foreign and just going to engulf it. And they don't care how big it is. All right, so let's talk a little bit about blood. It's going to be carrying, you know, essentially these cells. So white blood consists of plasma and formed elements, or your blood cells. Serum is a liquid portion of blood after a clot has formed, minus clotting factors. Right? Plasma is 92% water. It has, this is what passes out of your capillary system through diffusion. So water passes out, metabolic proteins, globulins. We want globulins to stay in your blood. These are big proteins, immunoglobulins, right? Clotting factors, hormones, and other chemicals and gases that support normal physiological functions. And once again, if you have a problem with your diabetic or you have a problem with your capillary bed to a specific area, usually distal extremities or skin or whatever, those cells can't get a lot of these things, all right? So their healing is completely diminished. You know, everyone's like, well, why do diabetics heal so slowly? Or, you know, why do their toes fall off? Or why do they have numbness? Because they have no blood supply left to the distal extremities because they've been non-compliant. They're flooding their bloodstream with glucose, which was pulling in fluid. They're just rupturing these capillary beds. Capillary beds are only one cell thick And they're really just transport and diffusion membranes. So they're really just got things in and out, and they're only, the lumen of that is only big enough to fit one red blood cell at a time, right? And that's why red blood cells have to be replaced every 120 days, right? Because they're going through the capillary beds and they're getting beat up constantly. By the time we get back to the spleen looks at it and is like, dude, you're all ragged, all right? So we're gonna just cleave you, recapture your heme and your globulin and your heme, and we're gonna replace you, right? Because remember, red blood cells don't have a nucleus, they can't reproduce. So we just, when they get all ragged, the lymphatic or the spleen goes in and destroys them. We just make more. The kidney produces erythropoietin, tells the bone marrow to make more red blood cells. Oh, here it is, hemopoiesis, production of blood cells. All right, so this is done by erythropoietin. And please realize, if you haven't been told this yet, I'm telling you now, hemopoiesis is production of blood cells. So that's white megakaryoblast platelets or red blood cells. Erythropoietin, erythro-red, is going to produce your red blood cells. All right, there's other enzymes, other poietins, and these are all produced in your kidney, and they send the signal to your bone marrow to produce these, as long as there's not some kind of aplastic disease going on. All right, your stem cells are undifferentiated cells, precursors of new blood cells. So we're going to go through your bloodlines here quickly. Remember, there's myoblasts and lymphoblasts. Right. So your lymphocytes, if you're not sure what those are your T cells and your B cells and your natural killer cells. Everything else is the other myocytes. Right. So leukocytes, once again, these are white blood cells, mature. Granulocytes have a lobe nucleus. If you're not sure what these are, the granulocytes are anything that ends in "phil," the phil family. So neutrophil, basophil, and eosinophil. They all have granules. When you looked at them in histology, they should have, like eosinophil should have been blue, neutrophil should have been very, very neutral. Basophils, eosinophil should have been pink. Basophil should have been slightly blue. Neutrophils should have been colorless. That's why they were neutral. They had no color, right? And they were named after the dye that they absorbed. Agranulocytes have no granules. These are unlobed. They have a rounded nucleus. All right, so here literally is your, is this going to work? Oops. Oh, so this doesn't expand out. Okay. All right, so here's your myeloid and your hemopoietic stem cell. So theoretically, this cell here can produce either bloodline. All right, as we go here, they become less and less differentiated. All right, all that I care that you know is your myeloid stem cells are going to make your erythroblasts, which are your red blood cells, megakaryoblast, right, which are make your platelets. Right. Myoblasts make the phil family, neutrophil, basophil, and eosinophil. All right, here's a monoblast, and I said before, monoblasts, when they leave, they turn into macrophages. And here are your lymphatic stem cells. So these are going to make your lymphoblasts. Blast is always non-mature. They're going to make your adult cells or your cytes, right? T cells, B cells. And everyone seems to forget about the natural killer cells. They're part of this bloodline too. So they will try, they'll definitely probably ask you something like this on your NCLEX or something. It's because a lot of people just forget it. So there it is. All right, and if we were talking about leukemia, all right, which is blood cancer or cancer of the blood, you only have two types. You have myeloid or lymphoid, and you either have acute or chronic, right? So there's only four. So as long as you know the myeloid, those are the cells that's affected, or if you know the lymphoid, is your lymphocytes, B cells and T cells. And here's the deal, if you don't want to memorize all that, and you probably, this is coming at you pretty fast, memorize one, the other ones are the other, right? So if you want to memorize the lymphoid, it's your lymphocytes, B cells, T cells, natural killer cells, everything else comes from the other blood, right? So neutrophils, these are your granulocytes. And I said these all end in phil. This is the phil family, granulocytes. These are lobe nuclei with lavender granules. These are phagocytes. And I said these are on, the first one's on call. They're usually elevated. You know, it's going to be a shift to the right, elevated for bacterial infection. And these go in and they'll just start eating and presenting. These are antigen presenting cells and they're phagocytes. Eosinophils or eosinophils. These are orange granules or pinkish. They're bilobed. These destroy eukaryotic pathogens, all right? So that being said, if somebody was not feeling well, we sent them in for blood work. You know, I said, well, Mrs. Jones, we have your blood work and your eosinophil count is elevated. You should only have 1 to 3% of your cells should be eosinophils, but you're up around the 8% right now. So then we're thinking some kind of eukaryotic pathogen. So I'm thinking Chinese liver fluke, could be some kind of protozoa, could be worm infection, something that's eukaryotic. These and eukaryotic pathogens are huge. These are big ass organisms. So these eosinophils literally, they go in and attach and they literally take chunks out of whatever it is trying to destroy it. So eosinophils generally elevated, it is a parasitic infection, or sometimes if you have an allergic reaction, they can elevate. Usually it's eukaryotic. Right? Basophils, all right, we'll make a distinction here. These are dark blue granules. Right? They release potent chemical mediators. All right, so basophils are in the blood. Mast cells are non-mobile elements bound to connective tissue. So these are laying in your connective tissue. All right, and these potent chemical mediators are things that incite allergic reaction. They release histamine, cytokinin, all these chemical things. So if you were bit or stung by something, you have a major allergic reaction. Your mast cells are in the tissue, connective tissue, so they're going to be triggered, and so are your basophils. All right. So please realize these are related cousins. They're very, very similar. Mast cells are almost permanently grounded in a certain area of your body, all over your body, connective tissue. Basophils are floating in your blood. All right, and they're almost non-existent. So I don't know if you had to learn never let monkeys eat bananas, or however you learned the stages of it. 60, 40, 830 should how they should go. Right? Never let monkeys eat bananas. So neutrophils, lymphocytes, monocytes, eosinophils, basophils, right? And then 60, 40, 830 is the percentage that they would go in. Right, so your lymphocytes, this is your bloodline. Right, so these are your lymphocytes are going to be your very specific learned response. So we'll learn about these and the adaptive immune system. Right? But a couple things I want to say today, and I'll repeat it over and over again at nauseum. Right? So they're about 20 to 35% of your white blood cells. These are very specific immune responses. These are not general. These are very specific to a certain antigen or a certain entity. So your B cells are humoral immunity. So humoral, back in the day, always meant blood or fluid. The humors from back in the day. Activated B cells produce antibodies. All right. That's actually not true. Does anybody know the right answer? Okay. B cells produce antibodies, or can produce antibodies, but B cells trigger plasma cells. Plasma cells actually produce antibodies. All right, so do not let anyone trick you with that. So if they ask you a question, B cells on there or plasma cells on there, theoretically, plasma cells make antibodies. All right, they're derived from B cells, but the correct answer is always going to be plasma cell. All right, and your T cells. These are cell-mediated immunity. All right. So here's the thing. If you can make notes or whatever, B cells always work in the blood. They're humoral in the blood. Okay. T cells are cell-mediated. These guys are literally hand-to-hand ninjas. All right. They don't send out antibodies. They don't send out anything. They literally go in, find the antigen, they lock onto it, and they kill it. This is cell-to-cell combat. Activated T cells modulate immune functions and kill foreign cells. So these guys are nasty. These are killer cells. CD8 cells are killer cells, and CD4 cells, we're going to learn, are the other T cells that are your helper cells. Those are the ones that are actually triage your entire immune function. All right. So anybody who has HIV or you've heard of AIDS or whatever, their CD4 cell count is very, very low. So they literally cannot triage their immune system. The CD4 cells are so low, and that's where the HIV virus likes the antigen-presenting cells, and it likes CD4 cells. So it goes in, replicates in these CD4 cells, and destroys them. That's how the virus works. So it kills all of their helper T cells, and it causes the immune system to be almost non-functional. We'll talk about that in a little bit. All right. Sorry, monocytes and macrophages. Remember, they're monocytes, adult cells in the blood. When they leave the blood and go to the tissue, interstitial fluid, they become macrophages or macrophages. And these are huge. These are the largest white blood cells, kidney-shaped nucleus. These are phagocytotic. Right, so these will go in and they'll engulf anything. And these things will eat bacteria and neutrophils until they literally die. They explode. All right. And then the cool thing they do, they leave this net as they die. They shoot out this net-like thing, and it traps a whole bunch of other bacteria. So it's a super cool process. Macrophages are final differentiation of monocytes when they leave the blood. And dendritic cells trap pathogens and participate in immune reactions. All right. So dendritic cells are those antigen-presenting cells in your endoreticular tissue that will grab the antigen, process it, get in your lymph vessels, and go to your lymph nodes and participate in immune reactions by presenting it to your immune system and say, listen, I just found this wherever. You need to make antibodies to it if you can. All right, so here's just a listing. Neutrophils, eosinophils, basophils, monocytes, and lymphocytes, what they do, and their percentage. All right, so I will be asking you a question. You know, your basophils are elevated. What could possibly be? All right, so generally this is going to be allergic reaction, allergic response right there, chemical mediators. Eosinophils, these are generally eukaryotic pathogens, worms or fungus. These are very large parasitic things. Neutrophils, general phagocytosis is usually elevated with a bacterial infection, something along those lines. All right, erythrocytes, I said, develop from bone marrow stem cells. They lose their nucleus. They're simple bicave sacs of hemoglobin. They lose their nucleus to carry more hemoglobin. We want them to be biconcave. We want them to be flexible. All right, so they're going to be like inner tube floating through your capillary bed, bending and twisting, getting through the tortuous capillary bed. Right? And they don't have a nucleus, so they can't replicate. So again, these are reproduced every 120 days. And remember, erythropoietin is going to be what is released by the kidneys when they realize you're hypoxic or anemic or something's going on. They realize that your red blood cell count is low or something's going on. They're going to pump out erythropoietin to produce more erythrocytes or red blood cells. Platelets are formed elements in circulating blood. They're not whole cells. These are derived from those megakaryoplasts. The megakaryoplasts literally go into your capillaries, and as the fluid goes by, it starts cleaving off little pieces of these megakaryoplasts, these great big cells, and they actually become little pieces of platelets, right? And those we know, or hopefully we know, are going to cause blood clotting. The other thing that I'm going to tell you guys, please remember, your blood should never clot in your bloodstream, ever. The minute it leaves your bloodstream, it should clot. So the minute your blood sees anything foreign, whether it is a needle, piece of glass, plastic, anything, it should clot. Once it's inside your blood, it should never clot. All right, we're going to find out with LPS and those gram-negative bacteria leaving that lipopolysaccharide. This will cause coagulated intravascular dissemination. So it will cause your platelets and red blood cells to start clotting inside your bloodstream. That can be very, very dangerous because then we start closing off capillaries and we decrease the blood supply to organs, organelles, arterioles, and whatever, kidneys. Kidneys are not happy when they don't get blood. So here's your lymphatic system, provides an auxiliary route for return of extracellular fluid to the circulatory system. So once again, these are going to be all around your capillary beds. So your capillaries, your arteries, veins, capillaries, and lymphatic system should all run together. And as that fluid's diffusing back and forth in the capillaries, remember, the hydrostatic pressure is always higher. It's going to push that fluid out of your capillary bed. Not all of it's going to go back into your capillary bed through oncotic pressure or albumin or whatever is pulling that pull force back in. So a certain percentage, usually 8 to 10%, is going to be staying in your interstitial fluid. Once again, when that fluid gets, pressure gets high, these lymph nodes or these lymph veins or venules, they open up and the fluid gets equalized. These go to percolate through your lymph nodes. So we can filter that out. And hopefully if there's any pathogenic agents in the interstitial fluid, lymph nodes are going to pick it up. It acts as a drain-off system for the inflammatory response. Remember, if we have an inflammatory response, it's being very protective. So your capillaries are going to open up. You know, they're one cell thick. They're going to open up, and a lot of that plasma we talked about earlier, that fluid, is going to leak out. Right? Okay, and then it builds up that pressure. We want that to flush out. If there's any toxins, we want to flush that out. And we want to build up that pressure. So if there is anything in the area, we want to bring it to the lymphatic system. We want to flush it through the lymph nodes. We want to filter it out and make sure there's anything pathogenic, we can build antibodies to it. So that's a very healthy response. Render surveillance, recognition, and protection against foreign material. So that's kind of its role. All right, the other thing that they may not have taught you, or maybe you don't remember, is your lymphatic system also has a digestive function where it actually can bring some of the lipids from your digestive system into your circulation, but that's separate than altogether. Lymphatic fluid, lymph is plasma-like liquid carried by lymphatic circulation. Form when blood components move out of the blood vessels into the extracellular space. And we do that with fever or infection or whatever. We kind of open up those capillary beds and we want that fluid to leave. Made up of water, dissolved salts, 2 to 5% proteins. Now, usually you want the proteins to stay in your blood, but we want those to leave, small proteins, in case we need them in the area for repair. We're assuming there's probably damage. We want it there, we want to deliver it. If it's not going to be used, we bring it back into circulation and we can use it somewhere else. We kind of deliver it there, drop it off. If you need it, use it. If not, we're going to recirculate. Transports white blood cells, fats, and cellular debris and infectious agents. So that's what it's going to do. Well, there it is, fats. That's part of your digestive system. You're going to bring those fats, cellular debris from, your macrophages just took on a bunch of bacteria or viruses or whatever. We don't want that laying around. We're going to bring it back, and then we're going to let the kidneys or liver or something else start getting rid of that. So here's your lymphatic vessels, lymphatic capillaries. So as they're moving towards your heart, they're going through all these collecting vessels. And we're hoping there's a B cell or T cell in there that's going to recognize something foreign and send out an antigen or antibody to it. Right? Just wanna make sure. All right, so it permeates all parts of the body except, remember, the central nervous system. We have that blood-brain barrier. We can't do it. The bone, placenta, and the thymus. Right, those are all sequestered. Thin walls easily permeated by extracellular fluid, which is then moved through the contraction of skeletal muscle. So I said it goes into here. As you move, it squeezes these. They're like one-way valves, like your veins. Moves everything, and it always moves towards your heart. Function to return lymph circulation flow is one direction towards the heart, eventually return to the bloodstream. Remember, light, the left thoracic duct, the right subclavian. Here's just a picture. You can follow these, you know, either 1, 2, 3, 4, A, B, C, D, however you're going to do it. Right? But we're going to just move that fluid through here. We want to percolate it. Now, the other thing I mentioned the other day was we have all of these afferent coming in here. We've only got one efferent leaving, so that's on purpose. We want all these to drain into here, this kidney-shaped lymph node. We literally want this fluid to percolate through all of these nodes. All right, we're going to slow it down because these B cells are just playing video games until they come across an antigen that they can make an antibody to. So we really want them to go through here. We'll give some time to percolate through there, have a reaction if they need to, and then we start bringing it back. Right. So your primary lymphoid organs are sites of lymphocytic origin and maturation. So the thymus and bone marrow. Right. So you know, if I say thymus, you guys should be thinking T cells immediately. Bone marrow would be your B cells. Right. Secondary lymphoid organs and tissues. These are circulatory-based locations, such as spleen and lymph nodes. Right. Collections of cells distributed through the body. All right, but the most important ones, believe it or not, are your skin and mucous membranes. Right, gastrointestinal associated lymphatic tissue. These other areas, these are in parts of your body. And think about this for a minute. These are all in areas that are literally outside your body. So we want these small vessels to be in your GI tract in case there's something in there we want exposed to. We want them in your respiratory tract. We want them in your groin, these areas distributed to the body in case there's something going on. Like Pyrus patches, I'm sure you guys have heard of those. I hope, right? Oh, there it is right there. All right. So thymus, high rate of growth and activity until puberty, then begins to shrink. It's a site of T cell maturation. So we said before, you have B cells and T cells. These are your lymphatic or lymphoid cell line. These are for your adaptive immune system. So these T cells are going to go to your thymus. Don't mix it up with your thyroid. They have to decide two things. Do they want to be a CD4 cell or a CD8 cell? They want to be a helper cell or killer cell. And they have to be able to distinguish cell from non-cell. So if we have a T cell that says, you know, I want to be a CD4 cell, okay, great. We send him out, or before he leaves the thymus. If it attacks one of your own cells, we have clonal deletion. We want to say, well, we don't want you, you failed that part of the exam. You can't tell self from non-self. So we're going to destroy that. So clonal deletion means that cell can't replicate. So we're going to delete that version of that CD4 cell, right? When we start talking at the end here about some of these autoimmune diseases, you know, there's theories where some of these T cells may leave and they're not quite 100% able to tell self and non-self. Right? The B cells leave your bloodstream already knowing what they're doing, but they're naive. So these are like, I don't want to say they're virgin B cells, but they're naive. They have all these antibody production areas in the B cell, but they have to be exposed to an antigen that triggers them. And we'll talk about that in a little bit. Right. So these lymph nodes, we said these are small encapsulated bean-shaped organs stationed along lymphatic channels and large blood vessels of the thoracic and abdominal cavities. And we saw a picture of that earlier. Now spleen, structurally similar to a lymph node. It's a great big lymph node. Filters circulating blood to remove worn out red blood cells and pathogens. And then we have the miscellaneous, the gall or gastrointestinal associated lymphatic tissue, or specifically Peyer's patches. So these are just kind of little tiny nodes spread out through the entire body so that we can, any pathogens we consume or that get into our external environment that try to get into the body, we hope that they'll be caught in one of these Peyer's patches. So we have to recognize things. We have an inflammatory response to flush it out, bring fluid, get the tissue to be inflamed, bring out some cytotoxic cytokine chemicals to alert the body what's going on. Then we want to go through phagocytosis. We want to go in cell eating or eating anything that's pathogenic, or some of these neutrophils and these blood cells are going to die doing their job. We have to go in and clean that up. You can't just leave it laying around. All right, the problem with this is when we go through phagocytosis, if we have something that's constantly irritating or constantly causing inflammation, every time we go in and clean it up, you have to remember too that we're cleaning up the area of damage, but we're always cleaning around the periphery a little bit. So constantly doing that, we start damaging tissue or areas or joints or parts of the body because we're constantly cleaning it up. And every time we clean it up, if we're constantly doing that, we start inflaming or tearing down the normal parenchyma of that area too. Interferon, we talked about a little bit. In complement, we're going to mention that a little bit. I'm not going to ask you guys certainly to remember the complement cascade. That's ridiculous. So I'm going to just give you two things I want you to remember. I want you to remember neutralization and opsonization. All right, so we can go in and we can literally, our immune system can super tag or tag a pathogen so it's much more, the immune system's much more capable of seeing it. It's blatantly obvious. Or we can go in and we can neutralize it. We can neutralize their receptor sites or their antigens or whatever. So it would be kind of like putting super glue on their receptors. We just kind of clog it up and neutralize it so they're useless. So inflammatory response, I'm going to mention this a few times, but remember, the inflammatory response is completely normal, and the inflammatory response is going to happen no matter what the damage is. I don't care whether it is a gunshot wound, laceration, burn, crush wound, brush burn, whatever it is, any damage whatsoever, there's always going to be an inflammatory response, and this is completely normal. It's annoying, but it's very, very useful. Right? So classic signs of symptoms are characterized by redness. So if you have inflammation, you're red. It's increased circulation and vasodilation in injured tissues in response to chemical mediators. So you're damaged, right? And these chemical mediators, in case you don't know, when you damage tissue, you break these cells. And when you break the cells, the cytoplasm opens up, right? So it gets into the interstitial fluid. Well, all the cells in your body know that, well, wait a minute, that cytoplasm, the contents of inside that cell should be inside that cell. So if I've got 100 cells that died from damage, all their cytoplasms are exploded and floating around. So all of that internal contents of the cell is floating around those cells. The body knows something's not right. It sends out these chemical mediators to tell the body, listen, I got damage here. I don't know what it is, but immediately I need you to increase circulation and vasodilation. We'll figure it out later, but I don't know what's going on. But right now, it kind of hits the red panic button. We'll deal with whatever it is, but send me redness immediately. Warmth is heat given off by the increased blood flow. Remember too that eventually we're going to trigger our hypothalamus to raise our temperature if we need to. And bringing heat to the area and blood flow is going to stimulate the immune system to rev up its activity. Now, swelling is increased fluid escaping into the tissue as blood vessels dilate. right? We've known it as edema. Right? So in that's going to be white blood cells, microbes, debris, necrotic tissue, and fluid collecting to form pus. So if you don't know what pus is, it is exudate or it is dead necrotic cells. Generally, it's white blood cells, but it can be bacteria, whatever is dead. So it's superfluous dead bacteria carcasses or white blood cell carcasses laying all over. All right, this is going to prevent the spread of infection. So we're going to cause swelling. We're going to compact that, make that high pressure. All right, so things can't really leave. We kind of bog it down with that until we can clean it up. In pain is stimulation of nerve endings. Please remember I mentioned this, I think 2 classes ago. Only your nerve fibers can generate pain. Remember that thalamus at its 90% of this. So if you have pain, usually there's swelling or damage, something is irritating or pushing on those afferent dendrites, sending that signal up to the cortex, telling you there's pain. So it's saying, you know, pay attention. Here's a major events and inflammation. Right, so, you know, we're just showing you, this is a nail, pin, or whatever. We've damaged tissue. We've actually punctured the tissue. So now we've gone through the first layer, or first line of defense. We've gone right through it. And now this needle is clearly infected with God knows what. And we've got bacteria. We punctured the skin. We're into the extracellular interstitial fluid. Now we've literally gone in, and these are really superficial. We've gone in, we've damaged that capillary bed. So that capillary bed is going to send out all kinds of signals. It's going to say, listen, I've damaged the endothelium. I need platelets. I need inflammation. I don't know what happened. We'll figure it out in a minute. But, you know, you're going to have your vascular reactions. So if you look here, we have vasodilation. Right? We're sending out neutrophils because they don't know what's going on. They're going to go in, start engulfing these bacteria, figure out what's going on. And the other thing, after we figure out the damage isn't life-threatening, this vasodilation is going to turn into vasoconstriction, so we're not pouring out blood if we can't fix it. Right? Here's edema and pus. So we're sending out all that fluid, all right, and the neutrophils are doing their job. They're containing this, they're cleaning it all up. It's all the debris, right? And then we have resolution, we have scar tissue forming, your capillary bed should heal. And then eventually we're going to send in macrophages to go in and just literally clean it up like a shop vac. We're going to go in and clean up all that area. All right, so here's some chemical mediators of the inflammatory system. And some of these you've probably heard of before. All right, so all that I want you to realize is we have vasoactive action. So we're going to cause vasodilation. And that's because we want to bring blood to the area in case we need supplies. Right? Over time, if we know it's not life-threatening, then we're going to want those to constrict. Right? But if you look, you guys have heard of histamine, serotonin, bradykinin causes major pain, so does prostaglandins. All right. And remember too, if you have a major inflammatory response, let's say you're allergic to something, you're going to take an antihistamine. We're going to say, listen, your response is correct. We want inflammation, but you're overreacting a little bit. I'm going to take a chemical to slow that down. Right. Initiating event, trauma, infection, necrosis, or foreign particle, neoplasm. These are going to produce mediators. All right, so these are going to cause vasoactive and chemotoxic effects. They're going to cause the blood vessels to dilate, but they're also going to call in these other elements that we might need. Cytokines such as interferon or interleukin, all right, to send out chemicals to warn cells. Arachidonic acid, metabolism, this is how Tylenol works. It stops arachidonic acid. We have platelet activators in case we want to repair the capillary bed. And then some chemotactic chemicals. So chemochemical tactic causing it to move. We're going to release these chemicals to cause things to move towards the area of pain. Remember, our cells can't send out Wi-Fi. They can't send out a text message. They don't have walkie-talkie cell phones. All they can do is if there's damage, they're going to send out chemical signals, and by diffusion gradients, these cells are going to go towards these chemicals. It's kind of a way of calling them in. Diapoesis, which means feet, migration of cells out of blood vessels into the tissue. So I said before that these capillary beds are one cell thick. When we need inflammation and we need to send things out, these literally open up. And this is diapoesis. They're like foot-like movement. These are going to literally leave. And here's the chemical. There's damage. So your cells send out these chemotoxic factors. Like I said, they don't have Wi-Fi or cell phones. They're sending out these chemicals. By chemical gradient, these are going to follow that signal. They're going to go towards the concentration of that, right? They follow the concentration gradient, and that's what calls them out. Right. So fever, anyone who's had it, it's really not pleasant. It's initiated by circulating pyro, like, what's that glassware? Whatever, pyrex. Yeah, pyrogens, which reset the hypothalamus to increase body temperature, signals muscles to increase heat production and vasoconstriction. Right, so fever, we could have a conversation, at what temperature do you lower the fever? When does it become dangerous to denature proteins or enzymes? 102, 103, 104. My thought process is if somebody has a low-grade fever, you just leave it alone. The body's doing what it wants. When it gets up to 101, 102, then we have to start thinking about, is it dangerous? We have to figure out what's going on, right? Exogenous. So exo is always out, produce products of infectious agents. So we're going to find out some of these staph and some of these strep bacteria are going to release exogenous pyrogens that are going to induce this fever, induce this red rash and things. Endogenous pyrogens are liberated by monocytes. So these are your cells, your grown monocytes that turn into macrophages, your neutrophils. Macrophages during phagocytosis. So if your macrophages are busy consuming a bunch of debris and dead bacteria, they're going to say, listen, you guys got to send out a signal. We got to raise the temperature. We're going to put the immune system on alert. All right. And we're hoping we'll raise the temperature so that these bacteria will slow down their replication. Send out some tumor necrotic factor in case there's cancerous tissue around. Interleukin-2 to signal a bunch of chemical processes. And the benefit of fever: inhibits multiplication of temperature-sensitive microorganisms, impedes nutrition of bacteria by reducing the available iron. So temperature, I said before, when we raise that temperature, your body is going to sequester iron and it sequesters zinc. And these are one of those cofactors when we talked about reproduction, transcription, translation. These bacteria need this to reproduce DNA. If we get rid of that, they won't have the supplies to reproduce. We can slow it down, right? But any of these bacteria that can produce hemolysins, all right, they can go in, they'll just go in and they'll cut a red blood cell, they'll lyse it, and they'll grab the heme right out of it. Right? Increases metabolism and stimulates the immune reactions and protective physiological processes. So it ramps up the activity of everything. So phagocytosis, these are big cells that eat. They survey tissue compartments and discover microbes, particulate matter. So that could be anything. It could be sliver, dirt, anything that's in a cutter, anything that's foreign. And dead or injured tissue, they're going to go in and clean it all up. They ingest and eliminate these materials, and they extract immunogenic information from foreign matter. So what they're saying here is these actually go in, they'll grab the antigens or any immunogenic information, anything from that bacteria or foreign substance that we can use to trigger our B cells to make antibodies. So any kind of markers, cell markers, receptors, pathogen-associated molecular patterns, anything that we can say is, look, I found this. I need to make an antibody to this because I found this. There's probably 10 million more of these somewhere. Right. And here, once again, we'll go over these are the phagocytes, or these are the cells that go in, they engulf or eat, and they hopefully will grab some of that immunogenic information and present it to your immune system. So neutrophils, as I said before, these are the most numerous, first on the scene. General purpose, they react early to bacterial and other foreign materials and to damaged tissue. And these can be phagocytes, and they can be antigen-presenting cells, or they're going to present that immunologic material to the immune system. So hopefully we can make antibodies to it. Eosinophils are attracted to sites of parasitic infections and antigen-antibody reactions. So any kind of allergic reaction, if they're elevated, we'd say, well, there's definitely an antigen-antibody reaction something. Your B cells are producing antibodies to some antigen, or we are infected with some kind of parasitic pathogen. Macrophages, once again, these are derived from monocytes. So monocytes leave the white blood, or they leave the bloodstream. They turn into macrophages. These scavenge and process foreign substances to prepare them for reactions with B and T lymphocytes. So macrophages, monocytes, and dendritic cells are your major players in presenting antigens, presenting foreign things to the immune system. So these are called antigen-presenting cells. So recognition of foreign cells. We have protein receptors within cell membranes of macrophages. They're called toll-like receptors. So this is a macrophage, a great big cell. It has these receptors. You know, look, they don't have eyes or scanners or, you know, they can't read Braille or whatever, but they have these receptors. They go around, they find something foreign, they attach to it. This literally turns on, you know, remember, if anything hits a receptor in your cell, it's going to send a signal to the nucleus to do something. There's something in the cell to do whatever that cell does. It turns it on or off. Like I can't reach up and turn the light switch on and off, but that's what they do. Whenever something signals or triggers a receptor, it's going to send information to the nucleus or the cell to do something. It's going to release cytokines, interleukins, inflammatory mediators. So this cell says, listen, I found this foreign material. I don't know what it is yet, but I know I want to send help. I'm going to send out a chemical signal to get more people here. Send out an interleukin to stimulate all the cells around me. And I want an inflammatory mediator just in case I need it. So chemotaxis and ingestion, phagocytes migrate and they recognize those pathogen-associated molecular patterns, and they go in and consume them. So phagolysomes. So these are lysomes. We talked about these earlier when we were talking about, in chapter 3, we were talking about eukaryotic cells and what these vesicles were, and I said they have some lysomes or digestive enzymes. So lysomes are fused with a phagosome, all right, definitely within 30 minutes. So if this phagosome goes in and eats it, we engulf it in this great big lysome vesicle. It's going to start digesting it or killing it in 30 minutes. And then destruction or elimination, all right? So here's, I said that oxygen's foreign to some bacteria. Some can deal with it. It's foreign to human respiratory cells if we can't deal with it either. So they can go in oxygen-dependent system. We can give it a respiratory burst. So they'll actually go in and burst it with oxygen. It's pretty ingenious, right? So if that bacteria can make catalase or superoxide dismutase or something like that, if we hit it with enough oxygen, it probably can't process all that. We'll end up killing it. It's another weapon we have in our arsenal. All right, liberation of lactic acid, lysomes, or nitric oxide. So we can hit it with a bunch of chemicals. If the lysomes don't work, the oxygen burst doesn't work, we'll hit it with lactic acid, lysomes, or nitric oxide. Hopefully we'll slow it down or kill it. So it's just showing what's going to engulf it. It's going to process it, all right? Lysome, digest it, process it. Take whatever we want, anything we can use, we're going to reuse again, right? And we're going to release the debris, right, into the interstitial fluid, And we're hoping that the lymphatic system will pick it up and we'll recirculate that and get it out of the area. All right, so interferon, I don't care that you guys know alpha, beta, or gamma. Just realize these are small protein produced by certain white blood cells and tissue cells, all right? And I said before, these are produced in response to viruses, RNA, immune products, and various antigens. They bind to cell surfaces and induce expression in antiviral proteins, inhibit expression of cancer genes. And what they'll do too, they'll bind these up so the virus can't lock on and get into the cell. Or a lot of times they'll send a signal and say, listen, you guys come across this viral component and it somehow gets inside your cell. What I want you to do is kill yourself. So you can't return to a viral factory. And complement, right? These are just blood proteins and they all kind of work like dominoes, right? And like I said, I don't care that you guys know any of this, but if you think about the classical pathway activated by the presence of antibody bound to a microorganism, so that's literally opsonization, classical pathway. Right? Or we can go in, and I'll show you in a minute. We can go in, classical pathway is just antigen antibody. So classically, we're just gonna stick proteins on this foreign thing and make sure our immune system recognizes it so they can't miss it. Skip off this. And we'll talk about this, the other cascade process next chapter. But the other thing that we can do too, we can do a membrane attack. So T cells literally do this. They go in, they attack the proteins, they punch a hole in the biphospholipid layer, and they literally expel a bunch of chemicals into the organism to kill it. They kind of basically rip open the cell membrane and they kind of puke in it, and then it kills the bacteria. It's kind of cool. I think that's going to be it for today, guys. All right, so what time is it now? Okay, 6:30. All right. All right, so I wanted here, this will be recorded. I'll pop this into Blackboard. All right, everyone's still here?
Yep.
Okay, I have a black screen. I have no idea what's going on. Okay,
Yeah, I'm
so,
here.
I'll record this. I'll download it into my computer. I'll upload it into Blackboard. This seemed to work okay for me. It was a little easier. I'll figure out some nuances with it. So I will see everyone, or most of you, on Thursday.
Yep.
All right, so
Sounds
if you
good.
get a chance, kind of pop in and check on that test. All right, I think it's 7 or 8 questions per chapter. Nothing I haven't gone over. And if it's something you don't remember, I gave you guys 70 minutes for 50 questions. So do what you know right away. And if you need to go look it up, look it up. All right, and let me know if there's a problem, okay?
Okay,
All right,
thanks.
guys, get
That's
some
good.
sleep. All
Thank
right,
you.
okay.
We'll
Right.
see you.
Dr. Phil.
Yeah, all right. Oh, there it is. Oh, now I see. Okay. I don't know what I did, but now I can see you guys.